Hello, everybody. This is Dr. Jim Daly at Daily Manufacturing, and I want to talk a little bit today about a product we don't have, but I'm thinking about, and probably will have. It's beta-alanine and slash L-carnosine. And it turns out that beta-alanine is used in the body to make L-carnosine. Now, don't get it confused with carnitine. They're two different things entirely. They have similar sounding names because they both were originally found in meat products. In fact, for both of them, that's the main dietary source of them. So that's what they have in common and why they have similar sounding names. They both come from the carne or carnivore or whatever root and have a similar sounding name. But it does get confusing to people because, even if I hear people talking, if I don't hear them well, I might get confused about what they're talking about because they are so similar. But they have very distinct functions in the body. And L-carnosine is not one that you hear a whole lot about, although it's certainly available and certainly out there, but it has some interesting properties. And that's what we're going to talk about today. And I'm still debating if we had the product, whether I just have the beta-alanine or whether I would actually combine it with L-carnosine or if I would want to have the two products separately. Um, and you'll kind of understand where I'm going with that as we get further along. So, with that said, Happy New Year, everybody, and let's get busy here and um, talk about our topic. And I'm going to share my screen here. And first of all, we'll get uh, this going. Now, I don't know how many of you are familiar with the Reams program Dr. Kerry Reams started that really got us started in business making products for his program. And one of the key things that they do is to measure pH in saliva and urine. And this kind of gives you a mirror of what's going on in the body to some extent, at least. And balancing that pH is an important part of it. And that's one of the reasons I kind of was focusing in on this because beta alanine and carnosine are known to be pH regulators in the body. And that's not really even something that's controversial. That's well documented, well understood. In fact, when I first started learning about it, I was reading about it in Guyton's textbook of medical physiology, about as mainstream of a source as you can get. So it's well established that it has this effect. And one other thing I'll point out here down on the screen, I'm talking about KISS science, keep it simple science. I'm trying to make it simple and find ways to discuss science with people that they can understand and yet get an appreciation for the science and what goes into it. It's a little bit tricky to do, but we'll keep working on that and trying. But I just use that KISS because you've heard it said, keep it simple, stupid, the acronym for KISS. Well, I'm not going to call people stupid or talk about that. I just say keep it simple science. So that's where I am with that, in case you're wondering. Now, when we look at carnosine, it's actually dipeptide. That means it's two kind of small amino acid-like materials um, combined. It's actually beta-alanine, which is an amino acid, and that's a picture of it over here. However, it's not an amino acid that forms proteins, really. It's not one of the amino acids that does that, but chemically it's an amino acid. but it does bind with L-histidine, which is an essential amino acid for making proteins. And um, that is makes L-carnosine when you put those two together. Now, unlike many peptides, carnitine is not really circulated systemically very well, but it's synthesized on site in the cells where it's used. Uh, High amounts of L-carnosine, which you're likely never to see unless you have a genetic problem, uh, can be toxic. Carnosine is actually being recognized now as a neurotransmitter, and it's neurotoxic in very high amounts. And uh, usually, when you see somebody that has a problem with that, it's because they have a deficiency in what's called carnosinase, which is an enzyme that breaks down L-carnosine. And it's pretty much of that in the body. And you don't have carnosine circulating in the body because there's so much carnosinase there that it just breaks it down almost immediately. So if you want to get carnosine into tissues, you basically have to take beta-alanine, which is the limiting factor for making carnosine in most cells. Um, so to get L-carnosine in the body, you take beta-alanine. One exception to that is in the gut. Carnosinase is not in the intestinal tract that much, and does seem to be some benefits in the intestinal system for L-carnosine. And so people do take L-carnosine. You'll see it sold on Amazon and online for gut health. So we'll talk about that a little bit more, but that's not the main part of our conversation today. Carnosine is a very important pH buffer in uh, the body, and particularly in the brain and in muscle tissue, that's skeletal muscle, heart, uh, both. And so it keeps you from getting overly acidic in those tissues, or for that matter, overly alkaline, although that's not usually the problem. But a buffer, by definition, can go both ways, and carnosine can. And if you get overly acidic, it brings it back more alkaline. If you get overly alkaline, it brings it back more into the proper range and makes it a little bit more acidic. It's found in both skeletal and cardiac muscles, like we sold, and is also high in the brain and nervous system tissues. Um, so L-carnosine functions as a biological regulator and contributes to tissue health and muscle function. Now, as we said, biosynthesis of carnosine is by enzymatically combining L-carnosine with the amino acid histidine in biological systems. Interesting, histidine can also be used to make histamine, which is important and has a lot of negative connotations out there because that's what's the problem a lot of times with your allergies and things like that, and other things. And one area of carnosine that really I need to look at more. I'm just sort of scratching the surface on that one. It does bind to the histamine receptor and seems like it might have some benefits there as far as perhaps blocking the effects of histamine itself. You know, it may have an antagonistic effect to the receptor, which means that it causes it not to work well instead of uh, having the effects of histamine that we all know about. So that's kind of an interesting thing, but not quite there yet on understanding how that all works. It's You seem to be getting a lot of varied information on that, and so I'm still trying to sort that one out. But the main thing we're interested in um, is the role of beta-alanine in making carnosine production, and that's what we usually think of it for. In fact, there's not very many things that beta-alanine does other than make other things. Um, the importance of histidine, along with beta-alanine, is really making L-carnosine. That's the probably the number one thing that we're going to see for that. Now, talk a little bit about buffering here and acidity. And acidity is just basically the concentration of hydrogen. There's several definitions of the Lowry definition. Uh, an acid is a proton or quote hydrogen ion donor, and acidity reflects how readily a substrate releases its protons. Arrhenius definition: an acid is a substance that increases the concentration of hydrogen H plus or H2O3, H3O2. I mean, that's has extra hydrogen in the water there. So increasing those concentrations. So what we're looking at anyway is basically a when you're overly acidic, an excess of hydrogen ions. Um, the role of biological buffers is to help maintain pH stability by reversibly binding hydrogen ions in body fluids. It's one of the major ways. Uh, and L-carnosine works in muscle cells to prevent excessive acidity during periods of metabolic stress. So it can grab onto some of that hydrogen and hold it where it's not if working and release it if it's needed. It can go both ways. And we know that in the blood, the pH is, in a range of 7.3 to 7.5. It's very tightly regulated. Interstitial fluid between cells in the body right at 7.4, so pretty much reflects what's in the blood. However, in intracellular fluid of muscle cells, it can be ranging from about 6.8 to 7.4, or it can get even more acidic than 6.8 in some cases, and that really starts limiting your ability to function well. So, how carnosine buffers hydrogen ions? And we'll just sort of briefly talk about this. Carnosine's imidazole ring dynamically accumulates and releases protons to regulate acidity in muscle tissue. So it binds to them and accumulates them or releases them. Uh, and that's how it basically works. This buffering action stabilizes pH during intense exercise, for instance, preventing harmful acid buildup in muscles. Um, Muscle function protection by mitigating these changes. L-carnosine helps to maintain optimal muscle function and performance under stress. And this is also important in cardiac tissues, although we see fewer studies on that. It does protect the heart as well. So, um, we'll keep on going. This is just kind of a little cartoon illustration of how that works. That's something that ChatGPT gave me. So, and you can see over here, you get beta-alanine, and it becomes L-carnosine. Exercise produces these lactic acids and things when you're doing exercise. Your pH drops, the carnosine binds to it and has a buffer reaction and neutralizes the uh, pH roughly and makes it more stable. So it reduces acidity and prevents fatigue. And over acidification is one of the mechanisms, whereas that occurs when you exercise a lot and you become fatigued and can't go on. Now, there's other things too. It's just not only being overly acidic and lactic acid buildup, but that's one of the things that is occurring. So it has a role in exercise in delaying muscle fatigue and improving performance. And that's due to its buffering mechanism. L-carnitine buffers pH changes in muscles, preventing acidity buildup. And by doing so, delays muscle fatigue, supports sustained endurance, and improved performance. And this would be something you would see mostly beneficial in endurance exercises, not so much in weightlifting because you don't really have a pH buildup when you're doing a few reps of so say weightlifting, bench presses, or whatever. So this would be more something that would be beneficial to runners or uh, tennis players, whoever, uh, who have to have a lot of endurance and that type of exercise. Now, and it has been shown that uh, it protects against acidosis in high intensity exercise, where you have lactic acid buildup. It accumulates in the muscles during intense exercise, causing discomfort and limiting performance. L-carnosine helps neutralize acid buildup in muscles and reduces acidosis and enhances muscle function. By mitigating acidosis, L-carnosine supports faster recovery and improved in performance in high intensity exercise. And this is a study from International Journal, a Journal of International Society for Sports Nutrition. And here it talks about beta-alanine is a well-established ergogenic aid that enhances muscle carnosine levels and buffering capacity during high intensity exercise. However, its role in improving strength and power performance remain inconsistent across the literature. This systemic review investigates whether dosing strategy rather than duration alone is the critical determinant in resistance trained populations. Conclusions, we'll just skip down to that. To optimize strength and power outcomes, beta-alanine supplementation should emphasize fragmented dosing protocols of 4 to 6.4 grams per day, sustained over a 5 to 8 weeks, particularly when implemented during training phases characterized by high metabolic stress. And what they found is that if you took a whole lot of the beta alanine at once, you really didn't get a whole lot of benefit. But if you took small doses throughout the day, it worked a whole lot better. And they couldn't even use a sustained release one and get the same effect as just taking the smaller amounts through the day. And probably that's because a lot of quote sustained release are really just delayed release. So, and they don't really sustain that well. Unfortunately, that's the case in very often. So, but we do see that there's very good evidence for using this for acidosis, at least when it's caused by um, exercise. Now, what I'm wondering is uh, how about acidosis caused by other things? There are physiological conditions other than just exercise that can cause acidosis. Does it help in those situations? And we're still kind of haven't found an answer to that question yet. How many? It has been shown in heart to be beneficial. It has been shown to be beneficial in the brain, but that's probably for a different mechanism, not just the buffering there, but the neurotransmitter um, part of it. But It also is a pretty good antioxidant, uh, and anti-glycation is something that it seems to present. And that may be, again, because it's adjusting the pH. The When you talk about anti-glycation effects, that's been shown in diabetics that carnosine prevents harmful glycation of proteins, uh, and that protects cellular protein from damage. Let's just back up for a little bit and define what glycation is. Glycation is a non-enzymatic, so it's uncontrolled binding of glucose to proteins. Now, you have glycation in diabetes because you have elevated glucose in the blood. That makes it easily done in an uncontrolled manner. So that's what glycation is, and it does it. Now, there is also something called glycosylation. That's totally different. Well, not totally different, but it's different. That's a controlled binding of glucose to proteins, and the glucose is bound into specific proteins and specific places on the protein, and that's a good thing usually. So glycation versus glycosylation. Uh, Glycation is uncontrolled and pretty much always harmful. Glycos Glycosylation is controlled and usually beneficial. And uh, the cellular protection is important in an anti-aging type product too, because by reducing oxidative damage and glycation, L-carnosine helps maintain cell function and delays age-related dysfunction. So that's what we're looking at in that regard. Uh, the neuroprotective effects, L-carnosine supports neurons. L-carnosine aids neuronal function by supporting cellular health and communication between neurons. Like I said, it's a neurotransmitter. I suspect it also has some of the pH buffering effects in the brain as well, because the brain does have quite a bit of it, and the brain often has a hard time moving things in and out well. So I suspect that carnosine, ooh, don't want to make that mistake, carnosine also helps protect the brain from becoming overly acidic at times. Um, but it also protects against these glycation reactions, which lead to neurodegeneration. And overall, it's been shown to be beneficial for cognitive uh, health in children. In that particular study, they didn't find it in adults so much. Uh, so it's been found to be beneficial in a number of different ways. Now, recommendations for beta-alanine supplementation, usually between 2 to 6 grams per day. That's quite a lot. And like we saw in the other study, it's better if you kind of spread that out during the day and not take it at once. Um, and that will be enough to see an increase in muscle carnosine. And there again, it works at best over a period of time. Uh, so, so here we're finding that carnosine is something that we can make in the body by giving beta-alanine. Beta-alanine supplementation is generally pretty safe uh, when people take it appropriately. Um, Sometimes people have taken too much and they have a mild paresthesia, which is tingling in the skin and stuff, but that's usually pretty harmless. And we haven't seen anything so far much worse than that um, by overdoing beta-alanine. But you still want to make sure you maintain a reasonable and proper dose, because the idea that more is always better is a bad idea. So getting it right is just as important for this as for anything else. And proper dosing helps minimize potential adverse effects. that could come up. So with that, I've sort of finished with L-carnosine. Um, it's an interesting product, and I'm still not quite sure. L-carnosine itself is generally sold for intestinal health, and it's been shown. Actually, it was developed for that about 30 years ago in Japan. Um, as a drug initially, but end up being sold mostly as a dietary supplement, but uh, for gut health, and it was for originally for peptic ulcers, is what they were trying to develop it for, and I think it's pretty well been shown to be at least reasonably effective for that, and for leaky gut syndrome, it's also seems to be effective for leaky gut. So we find a lot of really pretty good companies selling L-carnosine. Well, zinc carnosine is what they're really selling. That's what was Japan developed in Japan. But um, I've sort of debated whether I would want to make a product with the beta-alanine and the zinc carnosine combined. I'm not quite sure because they are used in different for different reasons, and there is overlap between the two. Um, but that's something I'm still sort of debating in my own mind. The doses are vastly different. So if you were going to give them, you would put much less, a small amount of zinc carnosine in with, much bigger dose of the beta-alanine. Um, but anyway, that's something I'm still debating. But it's a product that will probably come up pretty soon. I um, working out the details now and doses and things like that. But anybody have any comments or questions about it, or anybody happen to know much about it? Maybe I have a lot to learn yet, so I'm hoping be happy if somebody here knows something I don't know. Would anybody like to jump in and contribute or ask a question?
Hey, Jim. Yeah, I think interesting talk. I know beta-alanine for quite a while, but
Yeah.
didn't pay attention to L-carnitine.
Oh,
But
really?
I just want to add one thing about glycation that you mentioned.
Uh-huh.
So glycation, yes, reducing sugar can react with the protein that cause glycation. Um, but for most of the glycation are due to the accumulation of reactive carbonyl species.
Hmm.
These reactive RCS are more reactive than uh, reducing sugar, so that can cause the formation of ADEs, advanced glycation and the product, the
Right.
glycated protein. So my lab has been studying glycation for like almost 20 years. Yeah.
Okay. Um, well, you haven't done anything with L-carnosine on that.
No, we focus on dietary phyto, like a small molecular phytochemical,
Yeah.
mostly polyphenol flavonoids.
Okay.
Yeah.
Yeah. Well, L-carnosine is, a pretty small molecule itself. It's just histidine and beta-alanine.
Yeah,
Uh,
yeah.
but yeah, it's an interesting molecule. Yeah. Hopefully, well, I'm learning still about it, and also kind of interested in how it binds to what happens when it binds to the histidine receptor. The L-carnosine seems to be able to bind to that receptor, histamine receptor. So that's kind of something a direction I'm wanting to go a little bit too, and that could be a good thing or a bad thing. depending on that. But it appears from what I'm seeing so far, that's probably a good thing. Um, yeah, so yeah, this is an interesting topic, and um, I'm kind of interested in seeing where we go with it because we haven't gotten there yet. But I think it is something that's a really a viable product and certainly beneficial for certain situations. And for athletes, it's pretty well documented that it uh, gives you a bit of an edge if you're a runner, for instance. Yeah, so that's quite well documented. But it's well known as a physiological buffer. There again, that goes back to just basic textbooks like Guyton's medical physiology. Um, that's probably the uh, his medical physiology book is one of the really fundamental texts for medical students and physiology. So with that, anybody else have any questions? Okay, well, we'll uh, shut this down and Happy New Year, everybody. And I hope you're doing great. And we'll talk to you again next month. And in the meantime, I do post talk sometimes on Facebook. I posted one, what was it, last week or the week before? And I'll probably post one either this week or first part of next week again. I'm working on a talk to put on Facebook. So if you're not on Facebook and you might want to get on there and just be friends with me or with daily manufacturing and see what we put on there as well. But everyone have a wonderful evening, and we'll talk to you next month.
Thank you, Dr. Jim.
Thank you. Bye.
Uh, Talk to you soon. Yeah, and Shaman, we'll get together again soon and talk some more about product developments too. We've been talking about working on um, prebiotic product. So that's something we need to. I've been really interested in and don't have yet either. Okay. Well, goodbye,
Yes.
everyone.
Okay. Bye.
Bye.